Okay, good afternoon, everybody. I will be talking about molecular mechanisms of arrhythmia and sudden death in obesity. And arrhythmia refers to any electrical abnormality in the heart. So Abe has already talked about long QT interval, but obesity is associated with various surface changes on the electrocardiogram. There's also increased P-wave duration and dispersion as another common abnormality in these subjects. Obese people also have premature ventricular contractions, and this has been shown in several human studies and even in obese people with normal blood pressure. Obese individuals also have increased cardiac sudden death, and in fact, obesity-induced cardiomyopathy is believed to be the leading cause of non-ischemic sudden death today. And what's interesting is that this phenomena is not just limited to obese subjects, but people with diabetes and the metabolic syndrome. They also exhibit long QT and other EKG abnormalities. So Abe has already familiarized us with the EKG. I'm going to do it again. The top row here is a ventricular action potential, and the bottom row here is an EKG. The left-hand panel is a physiologically normal subject, and this is the long QT subject. And I'm going to start by talking about this panel right here of the ventricular action potential in a normal subject. So in order for the heart muscle to contract, you require a balance of sodium, potassium, and calcium currents. And these are regulated by the opening and closing of sodium channels, calcium channels, and potassium channels. So right here we have the upstroke of the action potential, which is marked by the opening of the sodium channels. And then, once we reach the peak, the cell is completely depolarized. It reaches a peak positive state. We have the opening of the potassium channels. And then we have the concurrent opening of calcium channels, which explains this plateau here before we see complete repolarization with the opening of the rest of the potassium channels. So potassium is obviously very important in the repolarization of the heart muscle. And you can see that this matches quite nicely with the EKG, which Abe was talking about. This describes the ventricular repolarization, and as we can see, the opening of the potassium channels marks with the repolarization here on the EKG. On the right-hand side, I just want to emphasize that this process is prolonged, and the repolarization takes much longer than it does in this panel. Similarly, the QT interval in this patient is longer than the QT interval in this patient. So we've developed a model to explain long QT and arrhythmia development. So obesity serves as some kind of stimulus. There's a change in the molecular pathway that we're currently investigating. The ion channels required for depolarization and repolarization are downregulated, and when they're downregulated, we can't generate the currents necessary for consistent depolarization and repolarization. And what we get is arrhythmia. And I just want to say, actually, that currently we have few therapeutic options to prevent sudden death and arrhythmia in these subjects. Currently, we rely on implanted cardioverters and defibrillators, but this is often limited and costly. So we're looking at the transcriptional regulation of the potassium channels, which are important in the repolarization of the heart muscle. However, for quite a long time, we didn't understand this system in the heart. We actually observed we studied it in neurons. So basically, there are these camp response elements, or Cre sites, in the potassium channel promoters. And CREB is a transcription factor. When phosphorylated, it becomes activated, and it binds these Cre sites and increases the transcription of genes downstream. I'm not going to discuss all of these genes. I'm going to only talk about the potassium channels. So obviously, potassium channels are very important nervous system function because you need to repolarize cells, and this is appreciated in the nervous system. the function of Krebs was appreciated in the nervous system, but not in the cardiac system. Until recently, in 2011, Michael Rosen, actually here at Columbia, discovered that reductions in Krebs lead to a reduction of a potassium channel in an artificial pacing model in dogs. The artificial pacing model is used to study arrhythmia. And that's when we got the idea that maybe we can look at Krebs and potassium channels in the heart to explain the long QT phenomena that's occurring. And this is actually a great example of how neuroscience has benefited cardiac research, because neurons and cardiac cells actually share a lot of similarities. They're both excitable cells. They both depend on ion channels to depolarize and repolarize, and they depend on the ion currents generated from these channels. We developed this hypothesis that if you have a reduction in Krebs, then you get a reduction in the expression of the potassium channels. And when you don't have the potassium channels, you have abnormalities in the ion currents, and they lead to long QT and arrhythmia. So this picture here shows the downregulation of Krebs, and then once you don't have Krebs binding, you get decreased expression of Kv 1.5, Kv 2.1. Those are just two ion channels. And then you get the development of long QT and arrhythmia that I've been discussing. So I have focused on two methods in particular. I'm doing promoter-reporter assays using the potassium promoter in cell lines. I'm using H9C2 cells, which are rat ventricular myoblasts. And I'm also doing mutagenesis experiments to identify the sites, the Cree sites, of exactly where the Krebs binds in the promoter region of these potassium channels. So this is a vector that we have generated tagging the potassium channel promoter to a luciferase construct. And basically what I want to emphasize on this slide is that the binding of Krebs to the potassium channel promoter induces the transcription of the luciferase gene, leading to luminescence. Therefore, the more Krebs binding you have and the more potassium channel promoter transcription you have, the more luminescence you get. And this is how we can measure the activity of Krebs and these potassium channels. So these are some of the results obtained. Let's start with panel A. So Kv 1.5 is the potassium channel promoter for mouse DNA. And compared to the control, which is pcDNA3, an empty DNA vector, we see a statistically significant increase in the transcription of the Kv 1.5 channel. We did not find any significant activity with the Kv 4.2 transcription, however, using these luciferase assays. And then in B, we're looking at mouse DNA again for the Kv 1.5 channel, and we see a dose response as we increase the concentration of Krebs from 20 to 40 to 60 nanograms. We see increased transcription of the Kv 1.5 channel. I'm not going to talk about panel C. That's the mutagenesis experiment which I was talking about. In panel D, we have the difference between panel B and panel D is that D is now looking at human Kv 1.5 transcription. And we see the same dose response that we saw in B, up to as much as 10-fold for 60 nanogram dose compared to the baseline pcDNA3. I just wanted to include these results as well, showing hERG is a very important potassium channel in the human heart for repolarization, and Krebs also increases the transcription of this channel. And in this slide here, we're looking at palmitate, which is used to simulate a model of obesity. And when we stimulated these H9C2 cells with palmitate and then assessed the Krebs levels, and compared to the control, the H9C2 expressed less Krebs, which would support our hypothesis that as you decrease the Krebs, you probably get a decrease in the potassium channels and subsequently the currents. And in panel B, this is just reaffirming the same thing. We see a decrease in the palmitate-treated cells with Krebs. So to come back to this model, we can use a simple explanation based on our preliminary results to explain what's happening. That stimulus in obesity could be the palmitate, which then leads to the Krebs degradation, which then leads to the potassium channels being down, the loss of repolarizing currents, and then arrhythmia. In summary, these results have shown that Krebs upregulates the transcription of several cardiac potassium genes, both in mouse and human proteins. The palmitate-treated cells have less Krebs, and we are performing additional research to identify upstream regulators of Krebs and get a better grip on what's happening. I would like to thank my mentor, Dr. John Morrow, who is here today. I'd like to thank Dr. Haiyan Huang, who also works in our lab. She's here. Vaibhav Amin, who has familiarized me with many of the lab techniques I've been using. Thank everyone in the IHN, and of course you guys for your patience and interest. Thank you.